So welcome everybody to the first Path Rounds of the new year. It is my pleasure to introduce our speaker today, Dr. J. Kizhakkedathu. I hope I got that right. Yeah.
Correct.
Who is a professor and Tier 1 Canada Research Chair in the Department of Pathology and Laboratory Medicine, and also a principal investigator at the Center for Blood Research and an associate member with the Department of Chemistry, the School of Biomedical Engineering at the University of British Columbia. Jay is a leading cutting-edge translational research, leads a cutting-edge translational research program to innovate materials and therapeutics that can treat and prevent thrombosis, bleeding, immunothrombosis, and infection, immunological rejection of organs and cells, and the protection of tissues and organs in inflammatory conditions. Uh, his research has been published in both fundamental science and clinical journals, including top journals in biomaterials, biotechnology, materials, translational medicine, and hematology, among many others, and several patents which form the foundation of a few companies. Jay has received numerous awards, including the Canadian Research Chair, including the Distinguished Achievement Award in Excellence and in Basic and Science Research from the Faculty of Medicine in 2022. He's been named as a Fellow of Biomaterial Science and Engineering in 2020 by the World Biomaterials Congress, and also received the UBC Killam Research Prize in 2017. So very looking forward to this. Thank you very much for doing this, Jay, and look forward to hearing more about your research.
Good. Thank you, Will. Thank you very much for the introduction, and thank you everyone for listening. And Happy New Year. And also, I would like to thank the department for giving this opportunity for presenting part of our work. Will, can you hear me well?
Yeah, can hear you great.
Okay, good. Okay, so today it's my pleasure to kind of introduce you to kind of a part of the work we are doing the lab, and it's mainly focusing on kind of blood coagulation and thrombosis aspect of my research. So disclosures, actually, some of this work I'm talking today is currently being developed by Biosindera.
So you can't see your, I don't know if anybody else can. I can't see your slides. Sorry.
You cannot see my slides?
I don't know. I can't. Can everybody else see them or? No, I can't see your slides. Hear you well. Can't see the slides.
I can't see
Okay.
slides. So maybe you should share.
Okay, I'll do it one more time. Let's see.
I could see him when I logged on, but
Okay. Let's go back. I know something happened.
oh, did we lose him? All right, we'll see if we can get him back here. Now we're all good. Perfect.
Okay. Okay, can you see my slides now?
But now we can't hear you.
Yes.
Can you hear me?
Yes, we can. Yeah.
Okay. Can you see my slides too?
Yes.
Yeah, we still can't. We can't hear you, Jay. I can at least. Can anybody else?
Yeah, I can hear.
I can hear Jay, yes.
Okay. Can I start?
Yeah, you're good.
Okay, good. Happy New Year, everyone. And it's my pleasure and thanks, Bill, for the introduction, and thanks the department for giving this opportunity to present part of my work in our lab. Today I'll be talking about one aspect of the work, which is on blood coagulation, and then how we can address blood coagulation and thrombosis using different types of like agents what we are developing in the lab. Before beginning, I have a disclosure to make. One of this couple of things what we're talking today is being developed by the company Biosynthera. So my lab is focused on kind of biomaterials and biomedical technologies to address unmet clinical needs. So the lab mainly focused on biomaterials in contacting with blood. Today I'll be talking about kind of two aspects of the work, including macromolecular therapeutics and blood compatible surfaces. And this many of this work, his work is been published. Actually, as you can see that actually some of the key publications are shown here, but I'll be showing you kind of like some of the previous work as well as kind of the current ongoing work and also some unpublished work. The team on this part of the work is started from a couple of postdocs and graduate students and currently being worked by Haifeng Ji and David and Jesse on working on this project. So what I'll be doing in next 45 to 50 minutes, actually, I will be looking at kind of like how we are targeting kind of like polyanions as a therapeutic target in developing new molecules or new approaches to prevent thrombosis. Not only in kind of like treating the thrombosis, but also kind of like developing new methods to treat thrombosis associated with medical devices. So why we are interested in thrombosis, actually, which is as we look at the literature, as we look at the clinical scenario currently, cardiovascular diseases are one of the leading causes of death in the world. And then thrombosis is one of the major pathology associated with this, including a stroke, as well as venous thromboembolism, as well as myocardial infarction or heart attack is all associated with thrombosis. And this is manifested as kind of either arterial thrombosis or a venous thrombosis, and this is being treated by anticoagulants and antithrombotic agents. And in addition to this, medical devices associated thrombosis is another major issue, and which is it's about kind of $400 billion industry globally, actually, like is affected by one aspect of other in terms of the device associated thrombosis. Although I said actually like this condition is being treated with anticoagulants and antithrombotic as shown here. There are many FDA-approved drugs, but still actually like it is this problem is prevalent, and we need kind of like better approaches or better treatment methods to treat these conditions. As you can see, actually there are few drugs actually are shown here, including heparins and other novel oral anticoagulants, and all these anticoagulants or antithrombotic agents are targeting either one pathway or other pathways of coagulation or inhibiting one of the coagulation factors. But because it is inhibiting the coagulation pathways, there's a potential for the bleeding risk associated with that, and all of these anticoagulants are associated with the bleeding risk too. So in addition to developing better antithrombotic agents, we also have to consider developing kind of agents actually which does not have kind of bleeding risk and which would help in treating these patients kind of effectively. So how we can develop new drugs in this space? Okay, what we have to consider, and what new approaches actually we can take it. So I'll show you a cartoon here. It's a slightly busy slide, but I'll walk you through it. And this is kind of like the current literature is suggesting that actually, like in addition to coagulation factors, we could potentially target biological polyanions. These are polyanionic molecules which are either secreted by the cells or kind of like present in our system, which are not essential part of the coagulation cascade, but still actually like it could augment coagulation. For example, And if you look at here, actually long chain polyphosphate or kind of short chain polyphosphate, long chain polyphosphate is secreted by the bacteria, and which is involved in kind of like coagulation. It can initiate coagulation, and it can lead to the thrombosis. And this is kind of particularly relevant in the case of like sepsis conditions or hyperinflammatory, bacterial infection conditions. In the case of short chain polyphosphate, which is secreted by platelets, and this is under certain conditions, and it could also activate coagulation, leading to blood coagulation and leading to blood clot formation. In addition to that, extracellular DNA and activated neutrophil extracellular traps, which are kind of like shown in case of like either cancer-associated thrombosis, or in the case of like hyperinflammatory conditions, and this assembly of the polyanions are also involved in coagulation or involved in activating coagulation, leading to thrombosis. But people, although it has been shown in the pathology, it is these molecules are indicated in coagulation and thrombosis, but it's kind of very difficult to develop molecules or targeting these molecules. People try to develop antibodies, but because these are ubiquitous molecules, actually it's very hard to develop kind of like very specific antibodies without side effects. And so like that leads to kind of like other approaches actually we need to take advantage of, and that is where actually like we took advantage of some of the polymers we are developing. So if you look at these polyanions, you could see these are the chemistry and chemical structures of these polyanions. You could see that heparins, which is not a procoagulant, which is an anticoagulant, but it is involved in treating coagulation. But all other polyanions, such as polyphosphate, extracellular DNA, circulating RNA, or extracellular neutrophil extracellular traps, all are kind of like can activate blood coagulation and are implicated in thrombosis in multiple analysis. And if you look at these polyanions, you can see that the arrangement of anions or anionic charges and these polyanions are different. So this will give us a kind of a tool or kind of an opportunity for us to develop chemical methods to target these molecules. So we are looking at in our work, we are looking at kind of like it's 5-6 kind of like polyanions, and which could be potentially used as kind of an approach to prevent thrombosis or prevent bleeding. So in the next few next while, actually I'll take you through kind of like 4 or 5 different stories, and which will give you kind of like how we approach this problem and how we develop these molecules to treat or kind of develop inhibitors or developing better devices. The first story is about kind of like developing polyphosphate inhibitors. And this work is done in collaboration with Jim Morrissey at the University of Michigan, and we have published extensively on this subject, and it's an ongoing project. So why polyphosphates are interested? Actually, this is a polyphosphate chemical structure, and polyphosphate released by the platelets are involved in clot formation in multiple pathways. And we have seen that kind of the people with inherent disorders, with the platelets does not have the polyphosphates are protected against thrombosis. Actually, that is telling us that actually like kind of inhibiting polyphosphate could be a potential pathway for preventing thrombosis. So in years back, we have developed a molecule called UHRA, and which is a